Welcome back friends, I am Rajdeep Acharya back again with another new lecture, and in this lecture we are going to talk about the ELISA test. The full form of ELISA is actually Enzyme Linked Immunosorbent Assay. So let's get started. So basically what is ELISA? ELISA is actually ELISA. Enzyme linked immunosorbent assay is actually a immunological technique. It is actually a immunological technique which actually we use to detect particular antibody or particular antigen from the biological sample, from the biological sample. So basically ELISA, I mean enzyme linked immunosorbent assay, is actually a immunological technique which actually we use to detect particular antigen or antibody from biological sample. And the basic principle of ELISA is, in ELISA test we use a antibody. In ELISA test we use a antibody which is actually linked with the enzyme. So we use a antibody which is actually linked with the enzyme. So basically in ELISA test we use antibody which is linked with the enzyme, I mean enzyme linked antibody, and this enzyme linked antibody react with a substrate. This enzyme linked antibody react with a substrate, and they react and they produce color. After the reaction they produce color. COLOUR. So basically the enzyme linked antibody will react with the substrate and they will produce color, and when the color will produce, it will indicate that the presence of the antibody or antigen in the biological sample. So this is actually, guys, the basic principle of ELISA. Now guys, we will be talking about the type of ELISA. So basically ELISA is actually of two types. I mean enzyme linked immunosorbent assay is actually of two types. One type is actually direct ELISA. So one type is actually direct ELISA or sandwich ELISA. So this is actually first type of ELISA. So basically in the direct ELISA or sandwich ELISA, this type of ELISA, I mean the direct ELISA, is actually used to determine a particular antigen which is actually present in the serum sample of patient. So basically in direct ELISA, a microtiter plate is actually used. This microtiter plate is actually 96 well polystyrene microtiter plate. So basically this is actually a well, this is guys, this is actually a well, this is actually a well of 96 microtiter plate, and this oil is actually coated some antibody in the surface, some antibody. So basically this oil is coated by some antibody on the surface. And guys, it is actually it is actually the serum sample of patient. It is actually the serum sample of patient. So it is actually the serum sample of patient. So basically, when we will add this serum sample in this oil, the antigen, the particular antigen which is actually present in the serum sample, it will attach with the surface antibody. So this is actually the antigen which is actually present in the serum sample of patient. And after that we will incubate it in 37 degrees Celsius. After that we will incubate it in 37 degrees Celsius, and now we will incubate in 37 degrees Celsius. After that we will attach our enzyme linked antibody. So after that we will attach, in the next step we will attach our enzyme linked antibody. So it is our enzyme linked antibody. It is our enzyme linked antibody which we will attach in this oil. So basically this enzyme linked antibody is actually designed for a particular antigen that actually we want to detect. So basically this enzyme linked antibody is designed in the lab so that we can actually detect this particular antigen from the serum sample. So now this enzyme linked antibody will attach on this antigen. We will attach on this antigen. Enzyme linked antibody will attach on this antigen. So it is actually our enzyme linked antibody, and then we will incubate it at 37 degrees Celsius. So now we will incubate it in 37 degrees Celsius. After that guys, we will use repeated wash, and in the next stage we will use repeated wash. And here actually we are doing repeated wash so that the unbound enzyme linked antibody will wash away from the oil. And in the next step we will add substrate. So in the next stage we will add substrate guys, and now we are adding our substrate. It is our substrate. So when we will add our substrate will react with the enzyme linked antibody, and basically after the reaction they will change the color of this oil. So basically they are producing a particular color in the well, and after when the color they will produce the color after the reaction, this color is actually indicating that a particular antigen is actually present in this well for which the enzyme linked antibody has been designed. And basically, if the reaction of enzyme linked antibody with the substrate does not occur, then they will not produce the color. So then we will conclude that this is not the particular antigen for which we have designed our enzyme linked antibody. So the color is produced, which indicates that the particular antigen of this patient serum sample is present for which we have designed our enzyme linked antibody in the lab. So basically, by the use of direct ELISA, we can actually guys, by using the direct ELISA, we can easily detect the antigen from the patient serum sample, guys. So this is actually the direct ELISA. I hope you guys have learned about what is actually direct ELISA. Now guys, we'll move to the indirect ELISA. So at first I have to move this. Now guys, we are moving to indirect ELISA. Indirect ELISA, which is actually type 2. Indirect ELISA. So in indirect ELISA, the indirect ELISA is actually we use to detect the presence of antibody from the patient serum sample. So the indirect ELISA is actually used to detect the presence of antibody from the patient serum sample. So suppose guys, suppose it is a well of 96 polystyrene microtiter plate. Suppose guys, it is actually a well, and in this case the surface of this well is actually coated with antigen. The surface is actually coated with antigen, and guys, it is actually the patient serum sample. It is actually the patient serum sample. It is actually the patient serum sample, and the patient, and in this case we are actually want to detect the antibody from the patient serum sample. That's why we have coated this wall with antigen. So now when we will put the patient serum sample in the well, we will get to see that the antibody from the patient serum sample, the antibody from the patient serum sample, will attach. And after the attachment, in the next step guys, in the next step we will use to attach our enzyme linked antibody. So now we will attach our enzyme linked antibody. So basically this enzyme linked antibody is specially designed in lab to determine the antibody from the patient serum sample. So now the enzyme linked antibody the enzyme linked antibody attached. The enzyme linked antibody is now attached. So it is actually the enzyme linked antibody. So it is actually the enzyme linked antibody. After the attachment, we will use to incubate it in 37 degrees Celsius, and then we will do repeated wash. Now we will do repeated wash, and after the repeated wash, the unbound enzyme linked antibody will wash away from the well, and then we will add substrate. Then we will add substrate, and when we will add the substrate, when we will add the substrate will react with the enzyme linked antibody, and they will change the color of the soil. That means they are producing color after the reaction of enzyme linked antibody with the substrate. So they are actually changing the color. So when they will change the color, this color is actually producing by the reaction of enzyme linked antibody with the substrate. So basically they are producing the color inside the well, which is indicating that the particular antibody is present in the well. So this is actually the indirect ELISA technique, guys. So I hope you guys have learned what is actually indirect ELISA, what is actually direct or sandwich ELISA, what is the basic principle of ELISA, and what is actually ELISA, I mean enzyme linked immunosorbent assay. So I hope the concept of ELISA is clear now, guys. So I hope you guys have learned something from this video. Like this video, share this video among your friends who are in need of this topic. Comment me about which type of lecture you guys want from me, so that I will make lecture on those topics, so that you will get help from me. And also subscribe this channel. Thank you so much, guys. Thank you.